Hi, welcome to the next episode of Cell-fie Science. I'm Jen, and I'm bringing you a quick burst of science from my cell phone. Today we're going to be talking about photosynthesis, which is why I have this bright light behind me. Photosynthesis is the process by which plants take light from the sunlight and change it into biological energy. But did you know that it actually requires two separate chemical reactions? That's right, what we think of as photosynthesis, light into sugar, takes two different reactions. The first is called the light-dependent reactions. In this reaction, sunlight is captured and turned into chemical energy in the form of ATP and NADPH, with oxygen as the byproduct, which, if you remember from our stomata video, is what plants breathe out. And on the other side is the light-independent reaction, sometimes called the Calvin cycle, in which the chemical energy from the light-dependent reactions is used to fix carbon dioxide into sugars. Now, plants have special organelles called chloroplasts where they perform photosynthesis. And I isolated a whole bunch of chloroplasts in this cuvette, which is why it looks so green. Because they're two separate reactions, you can actually measure photosynthesis with either one. Today we're going to be doing the Hill reaction, which is named after the man who discovered that this photosynthesis was indeed two separate chemical reactions. By taking a Hill reagent, which is anything that can accept an electron at the end of the light-dependent reactions, we can observe the amount of photosynthesis occurring. Today we're going to be using DCPIP, which has a blue coloration before it accepts any electrons. This is also sometimes marketed as vitamin C reagent, so you can buy that on Amazon or any other area. When the DCPIP accepts the electron or becomes reduced, it will turn colorless, so we can actually measure these colors. Today, we're going to be adding some chloroplast to our DCPIP. And I've isolated a whole bunch of chloroplasts to do this. So here we go. Take some chloroplast out. Add it to our Hill reagent. Now it's very important that you get this all mixed up, because if you have a blue layer and a green layer, then you're not getting that actual reaction. Now, I want you to observe the color versus the color of pure chlorophyll. We would expect, as the Hill reaction occurs, this will start turning this color, so it will get lighter in color. You can also add stuff like herbicides, which is what I've done to this cuvette, and then measure the amount of color change by using a spectrophotometer. Or you can do the eyeball test, and sometimes the eyeball test is a lot easier. So I have some that I prepared earlier because this process takes time, and this is a short burst of science. These guys have been sitting under this really bright light for about 20 minutes. One had herbicide and one did not. Based on what I showed you before, there we go. Based on what I showed you before, which one do you think had the herbicide? Did you guess the darker one? You're right. This is the one that only had DCPIP, and this one had DCPIP plus a herbicide called diuron. This herbicide decouples the light-dependent reactions. That means the electrons can no longer get from point A to point B and give them to the DCPIP. So no reaction occurs in this one. Now you can do this with all sorts of different stuff. For example, maybe you want to look at different light intensities. Put it closer to the light source. Or do you have different color LEDs? What would happen if you used blue or red or green? You could also use different compounds than diuron, but remember, if you're using any chemicals, be sure to follow their manufacturer instructions. Additionally, always wear your personal protective gear. Gloves and glasses are a must. And if you're a kid, be sure you have parental approval. I'll leave the recipes in the video description so that you can make up the various solutions to do this if you'd like to on your own. But for now, that's lights out for this episode of Cell-fie Science. Bye!